Welcome viewers. Today we are going to discuss about bacterial exotoxins. Now before I go into the details of exotoxins, let me first give you an importance of exotoxins in human diseases. In eastern part of India, we have heard of the term cholera, which is a dreaded disease which has taken an epidemic proportions once upon a time, and this is watery diarrhea leading to loss of electrolytes from the body and resulting sometimes even in death. Now, what is the cause of this cholera disease? The cause of the cholera disease is a soluble protein. The first description of the disease was done by Filippo Passini in 1854, who was a noted Italian anatomist, and later it was Robert Koch who first found out the organism which is Vibrio cholerae which caused the disease. It was in Kolkata where researchers by Shambhunath De found out the causal substance for cholera, which is the cholera toxin. It's a soluble protein which is secreted by these bacteria, and these leads to development of cholera. How it was found? Now, they first infected animals with live cultures of cholera, allowed them to grow in the media, removed the culture, and they used the same media to infect organisms. So they found out that there is something in the media which caused the cholera, and they found out it was a soluble protein. Similarly, for exotoxins for various other diseases was found out. So today we will discuss on different topics. For example, the nature of exotoxins, the infection process by the exotoxins, the classification of exotoxins, that is type 1, type 2 and type 3, we will go into the details of it. The control and synthesis and the release of these exotoxic proteins by the host organisms, and the treatment of exotoxin related diseases. And lastly, we will discuss about the toxin-antitoxin system. So coming to exotoxins, these are, as we discussed, small soluble molecules which are secreted out of the bacteria, and mostly gram-positive and also few gram-negative bacteria can secrete these exotoxins. Now, once these exotoxins are produced, they can enter the host cells by different routes. The first route is known as entry through receptor-mediated endocytosis, and what happens, as you are seeing in the figure, first once the bacteria or the microorganism comes in contact with the host which it is going to infect, it releases the toxin. These toxin attaches to the receptors. Now the receptors are internalized by receptor-mediated endocytosis. Once this receptor ligand complex reaches the endosomes, which are membrane-bound organelles within the cell, there is influx of H+ ions leading to change in pH of the endosomes. And in this acidic pH, the receptor and ligand get separated, and the toxin comes out into the cytosol, and it can modulate the different activities of the cell, and it will vary from different toxin to toxin. Another process is direct entry, and this is a slightly modified mechanism where the toxin directly binds to the receptor and it triggers a cascade of signaling events leading to development of disease condition. Now, if you ask me, are there any specific organism doing these things through different routes? Now, coming to it, we will consider that there are some organisms, for example, the toxin Diphtheria toxin, which is released by the corresponding microorganisms which cause diphtheria. They can enter through both these routes, that is direct entry or through receptor-mediated endocytosis, while there are some other toxins like toxins given by Pseudomonas which can enter through one of the routes. Now coming to the nature of these exotoxins, as we discussed, these are soluble proteins, small in size, but they can also form aggregates, and they can be enzymatic in nature, or they can be pore forming, they can be membrane degrading, and depending on them, there can be different types of classification. The classification which we will study today is based on its site of action and as well as its mode of action. Depending on that, we first divide it into type 1 or cell surface acting exotoxins, type 2 or extracellularly acting exotoxins, and type 3 or intracellular acting exotoxins. Now coming to the type 1, it is again classified into different types. The first is the superantigens. So if you ask me what are superantigens, these superantigens are small molecules which are produced by bacteria like Staphylococcus, Streptococcus or Yersinia or Mycoplasmas, which can create toxic shock. Now how do they do it? These superantigens go and bind to the MHT class 2 receptors which are present in the antigen presenting cell in the infected organism, and they mediate the interaction of this MHC class 2 with the T cell receptors, which can have a V alpha or the V beta or gamma chains, and due to which it leads to activation of T cell and correspondingly inflammation or various disease conditions can happen. So what are the diseases it causes? It causes chronic inflammation, it may cause food poisoning, it causes acute rheumatic fever, and especially in children, and this may lead to heart diseases, and as well as Kawasaki disease, which is first found in Japanese children, but it has been found elsewhere also in the world, where also heart diseases happen due to infection by superantigens. The second type of class 1 exotoxins are the heat-stable enterotoxins, which are produced by E. coli. Now, these enterotoxins are heat-stable because if you cook these proteins up at 100 degrees, still their activity remains. And once these binds to the G cells and they bind to a particular receptor, as you see guanylin receptors, and they are known as the STs, they increase the intracellular concentration of cyclic GMP, and it in turn leads to activation of a cascade of events leading to release of electrolytes from the cells. And as the electrolytes get released as well as water get released, it leads to diarrhea and it might even lead to death. The second type of exotoxins are the type 2 exotoxins. Now the type 2 exotoxins actually act outside the cells. That means they can act between two cells or in the extracellular regions, and they mostly are enzymatic in nature. So we will go through it one by one. The first is hyaluronidase. Hyaluronidase is an enzyme which breaks down hyaluronic acid to its corresponding components, and what happens, hyaluronic acid is a cementing components between the human cells. So if it breaks down, the integrity of the tissue get disturbed, and because of which bacteria like Staphylococcus, Clostridium, Streptococcus, which produce these types of enzyme, can lead to tissue degradations. And the activity of the hyaluronidase can be tested in the laboratory by first taking HA solution, which is the hyaluronic acid solution, then adding these toxins and see whether it fragments these or not, and then correspondingly adding a reagent which will give a particular color. So we can have a colorimetric based assay to test out whether the hyaluronidase is produced in those cells or produced by the organisms or not. Another type 2 or class 2 exotoxin is the coagulase. The coagulase is an enzyme which catalyzes the conversion of fibrinogen to fibrin, which results in clot formation. That means unwanted clot. It is produced by organisms like Staphylococcus or Yersinia, and because of this clot formation, there can be various external syndromes. For example, you can have rashes, you can have even life-threatening condition, provided you allow these microorganisms to go for a while. The next is fibrinolysin. So what is fibrinolysin? Fibrinolysin is an enzyme which catalyzes the conversion of plasminogen to fibrinolytic enzyme plasmin. And what it does, it also affects the clotting factors, and the bacteria which produces is a Streptococcus and Staphylococcus. So both these enzymes will affect the regular blood clotting mechanisms. Another type is the lipases. So what are lipases? Lipases are enzymes which hydrolyzes the esters of glycerol with preferably long fatty acid chains, and because it can hydrolyze it, it helps the bacteria to penetrate the tissue and leading to formation of different abscesses, leading to formation of rashes. As you can see in the figure, an abscess is formed due to production of these lipases, and bacteria like Bacillus, Pseudomonas can produce these types of enzymes which are exotoxic in nature. Another is IgA protease. Now, the importance of these bacteria which produce such protease is that they grow in a mucous membrane where the humidity or the mucus secretion happens, and it degrades the secretory IgA. If you remember, the IgA is given out by the host organism to kill these microbes. It can cut this IgA, and so it can evade the host immune mechanism to destroy it, and correspondingly it can spread its own infection. And organisms like Neisseria, Streptococcus can produce these types of IgA proteases and can lead to pathological conditions like periodontal diseases, dental plaque formation, even urinary tract infection. Another enzyme is collagenase. This enzyme catalyzes the degradation of collagen, and the organisms which produces is Clostridium, Streptomyces or Pseudomonas. And if you remember, collagen is an important component of our skin, or nails, or hairs. And if the collagen gets degraded, these nails or hair degradation can happen, and various skin infection can also happen. We next come to the enzymes which are still type 2 exotoxins, but they can damage the membrane. One of them are hemolysin. As the word tells you, hemolysins act on the red blood cell and can lead to destruction of the red blood cell, causing anemia. And as you can see in this figure, once hemolysin is added, or organisms which produce hemolysins like Staphylococcus or E. coli or Aeromonas is added to these cultures or to red blood cell, they can cause lysis of the red blood cell and can cause unwanted anemia. Another thing is leukocidins, which are pore-forming toxins which results in lysis of leukocytes, and Staphylococcus is one of the organisms which produce such exotoxin. Another important exotoxin are phospholipases. What are phospholipases? These are enzymes which attack any cell with a phospholipid. And if you remember, there are cells with phospholipids in the membrane. And gram-positive and gram-negative bacteria like Bacillus, Clostridium, Listeria, they can all produce phospholipases, different types of phospholipases, and can enter the host cells and cause pathogenesis. So we see that even exoenzymes which can form small pores after they get released from the bacteria can help in entry of the bacteria. And another important thing I should tell you at this juncture, that while they can help evade the host immune system, they can also colonize the host. They can lead to release of substances like perforins because of this cascade of reaction, which in turn can aid in the entry of these organisms. But these are not exotoxin. The exotoxin driven release of small molecules which are produced by the host cells, which itself get infected. So you can see how these exotoxin can manipulate the host cell's environment so that it can create infection. The third type of exotoxins, according to our classification, are the intracellular acting exotoxins. One of the most important of them is the diphtheria toxin. Diphtheria toxin is produced by the microorganism Corynebacterium diphtheriae. And as the name tells, it causes diphtheria, where cells get lysogenized with some strains of this bacteria, and these molecules have different subunits. The B subunit mediates the binding of this toxin to the human cell surface receptor, and the A subunit has the enzymatic activity. And what enzymatic activity? The enzyme is ADP ribosyl transferase. What it does, when it acts with humans EF2 plus NAD, it will lead to formation of ADPR, EF2 plus nicotinamide plus H plus ions. And what is the effect of this? Because of the presence of nicotinamide NH plus ion, it causes inhibition of protein synthesis, ultimately resulting to cell death. So, diphtheria toxin will cause cell death eventually. Another important toxin is Pseudomonas aeruginosa exotoxin A. Exotoxin A is the most toxic of these proteins secreted by Pseudomonas aeruginosa. It works in a similar fashion to diphtheria toxin and it can damage the epithelial cells. The exoenzyme S, which is also produced by Pseudomonas aeruginosa, can create epithelial cell death. Now, another important type of toxin, and we have just mentioned it previously in our discussion, is the cholera toxin produced by Vibrio cholerae. The cholera toxin has 5 B subunit and an A1 and A2 subunit. So what it does, when these subunits get attached to the host cells, what I mean by host is where it is going to infect. It will bind to the receptor, get internalized, react with the G protein and lead to the activity of adenylate cyclase, and correspondingly the amount of cAMP goes up, and these cAMP trigger a cascade of events due to which the electrolyte comes out of the cells accompanied by water, so it causes watery diarrhea. The labile Escherichia coli toxin, unlike the heat stables toxin, has a similar activity to that of the cholera toxin, and what it can create watery diarrhea. Another important exotoxin is the Bordetella pertussis toxin, better known as the pertussis toxin. Now, the pertussis toxin is associated with whooping cough syndrome. And one of the things which is observed, as you can see in the picture, this can lead to ciliary stasis. Usually the cilia moves and that keeps things going and that keeps preventing infection. But once the cilia get static, then the epithelial cells are exposed to various types of infection, and one of them is whooping cough. The same types of type 3 toxins are the Shiga toxin, which is produced by Shigella, and they actually prevent formation of different ribosome because they cleaves the 20S subunit of the ribosomal RNA, and due to which the protein synthesis is inhibited, and it leads to diarrhea, it leads to hemorrhagic colitis or even hemolytic uremic syndromes. Another important toxin is the anthrax toxin, and Bacillus anthracis is the causal organism, and it has several factors. The first is it has a protective antigen, it has an edema factor and a lethal factor. The edema factor and the lethal factor work in cohort to change the microenvironment of the infected cell, leading to necrosis of the cell, and one of the most important effect is dermal necrosis caused by Bacillus anthracis. Another important type of type 3 toxin is tetanus toxin, and we are more aware of tetanus caused by Clostridium tetani. It produces a zinc-dependent endopeptidase that cleaves synaptobrevin. Now I should give you a small overview of what I am talking about. Now these synaptobrevins are proteins which are present at the end of the nerve terminal, and this helps in events known as exo and endocytosis, which in turn control the release of neurotransmitter at the synapse. So if the function of such protein is affected, the neurotransmission in turn will be affected, and thereby we will affect synaptic transmission, and there will be various neural diseases which is associated with this. Similar is the case of botulinum toxin given out by Clostridium botulinum, but it also is an endopeptidase which inhibits presynaptic acetylcholine release because it works on these exocytotic proteins, and correspondingly it may lead to muscle fatigue or even paralysis because synaptic transmission or neural transmission does not happen. The last of this category of type 3 is the procyanin. Procyanin is given out by Pseudomonas aeruginosa, which produces a non-enzyme protein, and this, because of its redox property, can affect the cascade of energy events happening during energy production in our body. Correspondingly, the energy synthesis mechanism is affected, leading to lethality and cell death. So what we have learned till now is the different classification of toxins. Now, if we ask, are the toxins just released by any bacteria when they come to the host? The answer is no. There are favorable conditions only when the exotoxins will be released. For example, if we talk of diphtheria, when the bacteria come and bind to the host cells, depending on the iron present on the microenvironment, it will release the diphtheria toxin or not. If the iron content is high, it will prevent the production of the diphtheria toxin and its corresponding release. Similarly, expression of cholera toxin and its related virulence is controlled by environmental osmolality and temperature. At very low temperature, there will be no effect. Now, if you ask me, are there any treatment of these diseases? I will first tell you, yes, there are treatments. We will think of these treatments on two aspects. First is symptom-based treatments. That is, there are symptoms which are given out by the body and we treat these symptoms, and mostly antibiotic treatment is preferred. But it has been observed that most of these bacteria nowadays are antibiotic resistant. And for this reason, if we want to kill the cause at the very root, then we should move out from antibody treatment and focus on neutralizing protein. What I mean by that, people have engineered the T cell receptor in such a way that these will catch up the exotoxins but will not get activated. That means it will prevent activation of the cascade of event which normally happens because some alteration of the receptor has been done, and this is neutralizing the effects of the toxin. And we are talking of one example. There is research going on nowadays to carry out these things, neutralizing the exotoxin effect at various levels. And what it does is, the aim of such research is to prevent the body's aggressive response to the presence of such exotoxins. Now, the discussion about exotoxin is incomplete without a discussion of the toxin-antitoxin system. So, what is it? Now, within the microorganism, these toxins are always produced. Now, if the toxins are produced, they can adversely affect the microorganism or the nearby environment, which may be already favorable to the microorganism. So, the microorganisms, that means the bacteria in this case, have developed a technique by means of which it produces antitoxins at the mRNA level so that the formation of the toxin protein is prevented. So, we have a toxin-antitoxin loci which usually prevents the formation of toxins. Under favorable condition, the antitoxin is not produced, that is the mRNA for the antitoxin is not produced, due to which there is corresponding translation of the mRNA of the toxin, leading to formation of these soluble proteins which are given out. So we see the importance of exotoxin in human diseases, and we also should acknowledge the fact that these soluble proteins are not just given out like that. They are given out by these microbes or by these bacteria, to be very specific, under favorable condition so that it can infect, colonize a host and obtain nutrients, and this colonization may lead to disease condition in human as well as in other animals. So today we have discussed about exotoxins, a discovery of cholera toxin to start with. Then we have discussed about the nature of exotoxins. We have gone through the classification based on its mode of action and site of action. And we have also discussed about the possible therapy and a toxin-antitoxin system, which is so very important for regulated release of the toxins. Thank you for your attention.